morning, sir. Good morning, my fellow students.
Good
My
morning.
name is Badmus Aishat, and I'm here to present my seminar work on course titled Dosage Form Evaluation and Drug Stability. And the title of my presentation is Principles and Importance of Product Stability Testing. Abstract. Product stability testing is a cornerstone in pharmaceutical development, ensuring that drug products restrain their intended physical, chemical, microbiological, therapeutic, and toxicological specifications throughout their shelf life. Stability testing provides physical data that supports the determination of expiration dates, optimal conditions, and appropriate packaging requirements. This seminar work dives into the principles guiding stability testing, including the types of stability studies, the methodologies involved, and the regulatory frameworks underlying the process. Moreover, the significance of stability testing is maintaining drug efficacy, ensuring patient safety, and meeting regulatory compliance is thoroughly discussed. An understanding of product stability testing is vital for pharmaceutical profession as it ensures the consistent delivery of high quality and safe medications to patients. The introduction. In the realm of pharmaceutical sciences, the stability of a drug product is of paramount importance. Stability refers to the ability of a pharmaceutical product to maintain its identity, strength, quality, and purity throughout the design shelf life. A stable product ensures that patients receive medications that are safe, effective, and reliable. Conversely, instability can result in the loss of therapeutic efficacy, the formation of toxic degradable products, or other harmful consequences. Product stability testing is therefore an essential procedure in drug development and commercialization. It involves systematic studies designed to understand how the quality of a drug varies with time under the influence of environmental factors such as temperature, humidity, and light. The results of these studies are pivotal in determining the recommended storage conditions, expiration dates, packaging and requirements for pharmaceutical products. Beyond the pharmaceutical industry, regulatory authorities worldwide, including the United States Food and Drug Administration (FDA) and the European Medicines Agency (EMA, emphasize the necessity of comprehensive stability testing. These agencies provide stringent guidelines that manufacturers must follow to ensure that products released into the market meet required safety and efficacy standards throughout their life cycle. This seminar presentation seeks to discuss in details the underlying principles of product stability testing, the various types of stability studies employed, the importance of selecting appropriate testing methodologies, and the broader significance of these practices in pharmaceutical quality assurance. A thorough understanding of stability testing is indispensable for pharmacists, researchers, manufacturers, and regulatory bodies aiming to uphold the highest standard pharmaceutical care. Now, definition and objectives of stability testing. Stability testing is a crucial process that evaluates the longevity and integrity of pharmaceutical products under controlled environmental conditions. According to the International Council of Harmonization (ICH) guidelines, stability testing involves exposing a drug product to conditions such as temperature, humidity, light, and physical stress to stimulate real-world storage and handling conditions over time. This testing provides essential data that ensures the product will maintain its identity, potency, and safety when used within its defined shelf life. The objectives of stability testing are multifaceted and include the following. 1. Determining shelf life. One of the primary objectives of stability testing is to determine the shelf life of a drug product. This includes identifying how long the product will retain its efficacy and the safety under specified storage conditions. The shelf life is typically determined by the degradation rate of the active pharmaceutical ingredients (API) and its metabolites. This is calculated using the data obtained from various stress conditions such as accelerated and real-time testing. Then we have establishing storage conditions. Stability testing helps identify the optimal storage conditions, including temperature, humidity, light exposure. Drug products must be tested under a variety of conditions to determine how environmental factors impact their stability. For instance, some products may degrade faster when exposed to high temperature or humidity, while others may be sensitive to light. Establishing packaging requirements. Stability testing also evaluates the suitability of packaging materials. The interaction between the drug product and its packaging is vital, as certain materials can affect the chemical stability of drug. For example, light sensitivity drug may require amber-colored bottles to prevent degradation due to exposure to light, while moisture-sensitive formulation may need desiccant packs. Number 4, ensuring regulatory compliance. Stability testing is essential for ensuring that drug products meet the standards required by the regulatory agency, such as FDA, EMA, and World Health Organization. These organizations mandate that pharmaceutical products undergo thorough stability testing to guarantee that they are safe and effective for public use throughout their shelf life. In summary, the primary goal of stability testing is to ensure the safety, efficacy, and quality of pharmaceutical products by understanding how they change over time under varying environmental conditions. Without stability testing, there would be no assurance that drugs are safe for use, effective, and properly stored throughout their market life. The principles of stability testing. Stability testing is grounded in several key principles, each designed to address different aspects of how a drug product can maintain its integrity and functionality throughout its shelf life. These principles are critical for evaluating the physical, chemical, and microbiological properties of pharmaceutical products under stress conditions. The major principles of stability testing include 1. Scientific methodology and design. Stability testing must be conducted using rigorous scientific methods. It involves using a well-defined set of conditions and parameters to replicate realistic storage scenarios. The design typically includes accelerated stability studies, subjecting the product to extreme conditions to hasten degradation, and real-time studies, observing the product under normal storage conditions over an extended period of time. 2. Time and temperature dependence. Stability degradation often follow a time-dependent pattern. Most pharmaceutical formulations degrade over time, but at varying rates depending on the environmental condition they are exposed to. The principles of Arrhenius law is often applied to understand how temperature influences chemical reactions and the degradation of drugs. The rate of degradation usually double for every 10 degrees Celsius rise in temperature, which is why accelerated studies at high temperature are commonly employed in stability testing. We have the environmental conditions. The stability of a drug is sensitive to environmental conditions like temperature, humidity, and light. High temperatures can increase the rate of chemical reaction, leading to faster degradation. Then we have physical, chemical, and microbiological properties. The principle of evaluating physical properties such as appearance, texture, color, and phase separation ensures that the product remains intact throughout its shelf life. Chemical stability is assessed by measuring changes in the concentration of the active ingredients or the formation of degradation products. For solid dosage form, Stability testing will check for changes in dissolution characteristics, which can directly affect the bioavailability of the drug. Finally, microbiological stability is a key consideration for sterile products and biologics, ensuring that no microbial growth occurs during the storage. Then we have the statistical analysis. Stability testing relies heavily on statistical analysis to interpret the data. Statistical tools are used to predict the shelf life, assess the stability profile of the drug, and understand how different conditions might affect the stability of the drug. The use of probabilistic modeling helps to predict future performance based on observed degradation rates, which is vital for establishing expiry dates. Factors affecting product stability. Various factors influence the stability of pharmaceutical products. Understanding these factors is vital for developing and maintaining safe and effective medications. The main factors that impact product stability include: 1. Temperature. Temperature is one of the most significant factors affecting the stability of drug products. High temperatures can accelerate to the degradation of both the active ingredients and excipients, leading to reduced potency and the formation of potentially harmful degradation products. Conversely, some drugs may crystallize or undergo other changes when exposed to low temperature. Therefore, the proper temperature range for storage is critical to ensuring product stability. We have the humidity. Moisture is another critical factor affecting stability, particularly for solid dosage form like tablets and capsules. High humidity can lead to the hydrolysis of the active ingredients, resulting in reduced efficacy and harmful byproducts. Certain excipients in the formulation may also absorb moisture, leading to physical changes such as clumping or loss of texture. Therefore, stability studies at varying level of humidity helps to determine the optimal storage condition for moisture-sensitive products. Light exposure. Light exposure-induced degradation is a significant concern for certain drugs that are sensitive to light. Photodegradation can alter the chemical structure of the drug, leading to loss of potency or the creation of toxic byproducts. Drugs like vitamins, e.g., vitamin D, or antibiotics like tetracycline, are known to be particularly susceptible to photodegradation. Stability testing under different light conditions, like under fluorescent lights, UV lights, helps in determining the appropriate packaging for such drugs, such as opaque or amber containers. Oxygen exposure. The presence of oxygen can lead to oxidation reaction, which may degrade the active ingredients in some drug formulations. Oxidation can result in loss of drug potency, change in color, or formation of potentially harmful byproducts. Drugs susceptible to oxidation may require packaging in airtight containers or with additions of stabilizer or prevent oxygen exposure. Then we have the pH level. The pH of the environment can also affect drug stability. Some drugs may degrade more rapidly under acidic or alkalinic condition. For example, some antibiotics like penicillin, they are unstable in acidic environment, while others may be unstable in basic conditions. Testing the stability of drug across a range of pH levels allow manufacturers to optimize formulation and adjust for varying conditions in different regions or climates. Now we have types of stability studies. Stability studies are categorized based on the testing conditions, the type of pharmaceutical product, and the purpose of the study. The two most common types of stability studies in pharmaceutical testing are accelerated stability studies and real-time stability studies, although there are also other specialized studies, including long-term studies and stress testing. Each type serves a unique purpose in ensuring that drug products meet the required quality standard throughout their shelf life. Now we're going to be discussing about the accelerated stability studies. Accelerated stability studies involve subjecting a drug product to extreme conditions such as high temperature, humidity, or light exposure in order to expedite the degradation process. These studies are conducted over a shorter period, often months, and are used to predict how a drug will behave under normal storage conditions over a longer time. For instance, in an accelerated study, a product might be stored at 40 degrees Celsius and 75% relative humidity for 6 months, with the result extrapolated to estimate the stability over the typical shelf life of the product. The primary advantage of accelerated studies is the ability to generate data more quickly, which is especially beneficial for drugs that are near the end of their development life cycle, or for products requiring urgent market approval. However, it is important to note that accelerated testing does not always perfectly replicate real-world conditions, and degradation behavior observed under these extreme conditions may not always be entirely accurate when applied to real-time situations. The real-time stability studies. In contrast to accelerated studies, real-time stability studies involve testing a drug product under conditions that mimic typical storage environments. This type of study is conducted over a much longer period, typically 12 months or more, to monitor the product performance and stability under realistic conditions. The primary goal is to understand how the product performs in its typical storage conditions and to establish its shelf life and expiration dates. Real-time studies are essential for providing conclusive evidence of a drug stability. These studies are conducted according to good manufacturing practices and can provide long-term data that demonstrate the drug product's safety, efficacy, and integrity over time. Then we have the long-term stability studies. Long-term stability studies are typically an extension of real-time stability studies conducted over an extended period, often 3 to 5 years, to gather comprehensive data about the stability of a product over its entire shelf life. These studies are important for ensuring the products maintain their efficacy and safety throughout the duration of their market availability, especially when subjected to routine storage conditions over time. Then the stress testing. Stress testing is designed to assess the stability of a drug product under extreme conditions that may not be encountered during normal use. This type of study is used to determine the stability of a product when exposed to factors like high heat, light, or humidity, as well as to understand how the product degrades under stress, like oxidation or hydrolysis. Stress testing is particularly important for identifying degradation pathways and for determining the potential breakdown products of the drug. These insights are essential in events of unforeseen circumstances or when developing alternative formulations. Then last one, which is in-use stability studies. In-use stability studies are conducted to evaluate the stability of a product once it has been opened and being used by the patient. These studies are important for products that are subject to multiple uses, such as liquid formulations, cream, and injectable products. This type of stability study ensures that the product remains stable and effective during the course of its usage and help define the appropriate duration for use after opening. In conclusion, on type of stability testing, the choice of stability study types depends on several factors, including the nature of the product, the development phase, regulatory requirements, and the intended market. Each type of stability study provides valuable information that help ensure that drug products are safe, effective, and reliable throughout their shelf life. A combination of accelerated, real-time, and long-term stability testing is often used to confirm the robustness of the pharmaceutical product. Methods and techniques for stability testing. Stability testing involves a variety of methods and techniques to assess the physical, chemical, and microbiological properties of pharmaceutical products. These methods ensure that The product remains safe, effective, and of high quality throughout its shelf life. Below are the key techniques. Number one, we have the physical testing methods. Physical testing assesses the drug's product appearance, texture, and other sensory attributes that might change over time. This includes the visual inspection. This is a simple yet crucial test where the appearance of product is examined for any physical changes such as color alteration, phase separation, and crystallization. For example, a suspension might form visible precipitates, or a tablet might become discolored, indicating degradation. We also have the solution testing under physical testing. For solid oral dosage forms like tablets or capsules, the solution testing measures how well the active pharmaceutical ingredient is released from the dosage form. Change in the solution characteristics over time can indicate a degradation in product performance. We also have particle size distribution. For suspensions or powder, particle size distribution can change due to aggregation or degradation. Monitoring particle size ensures the product stability, physical stability, and bioavailability, as smaller particles may absorb differently than larger ones. Then we have the viscosity measurement. For liquid formulation, viscosity can change as a result of chemical breakdown or interactions with excipients. Measuring viscosity over time helps detect changes in product formulation and ensures consistency in delivery of the drug. Then we have second, the chemical testing method. The chemical testing assesses the stability of the drug's active ingredients and excipients, focusing on the changes in their chemical composition. The following techniques are commonly used: the high performance liquid chromatography. It is one of the most widely used methods for chemical analysis in stability testing. It allows for the quantification of the active pharmaceutical ingredients and the detection of any degradation product by comparing the amount of API in a sample over time. It's possible to determine the rate of degradation and estimate the shelf life of the product. The gas chromatography, similar to high performance liquid chromatography, it is used for volatile substances and is useful in determining degradation by products that may evaporate or undergo phase changes. Gas chromatography can be used for analyzing substances such as alcohol, solvents, or gases produced during degradation. We have the spectroscopic method. Various spectroscopic techniques include ultraviolet spectroscopy, infrared spectroscopy, and nuclear magnetic resonance. They are methods to provide insights into the molecular structure of the drug and any potential degradation products. UV spectroscopy is particularly useful for drugs with chromophores, light-absorbing groups. Then we have the titration. Titration is used to measure the concentration of active ingredients in solid or liquid formulations. It is often employed to determine the extent of degradation due to hydrolysis or oxidation. Then we have the microbiological testing method. For sterile products or products that contain preservatives, microbiology testing is critical in stability studies to ensure the products remain free from microbial contamination. And these methods include the microbial challenge test. This test evaluates the ability of the product's preservative system to prevent the growth of bacteria, fungal, and other microorganisms by inoculating a product with known quantities of microbes and testing for their growth over time. Manufacturers can ensure that the product maintain its sterility. Then we have the endotoxin testing. For injectable products, especially biologics, endotoxin released from bacterial cells, endotoxin, which are toxins released from bacterial cells, their major concern. The Limulus amebocyte lysate test is used to detect endotoxins in drug products to ensure they do not exceed safety limits. Then you have the sterility testing. Sterility testing is essential for parenteral products, ophthalmic preparation, and other formulations that are intended to be sterile. The test ensures that no viable microorganisms are present in the drug product, which could otherwise lead to infections or other serious health issues. We have number 4 to be stability indicating methods. Stability indicating methods are techniques designed to assess changes in the product that directly relate to its stability. These methods can identify degradation products that might not be visible or might form only under certain conditions. Common stability indicating methods include stress testing and forced degradation studies. Then statistical analysis in stability testing. Statistical analysis play a significant role in interpreting data stability. The goal is to provide accurate predictions regarding the product's shelf life based on data collected from stability studies. Statistical tools help estimate the rate of degradation and predict the expiry date using models such as zero order, first order, and second order degradation kinetics. These models describe how the concentration of the active ingredient decreases over time under certain conditions. Accelerated stability models. These models use data from accelerated testing to predict long-term stability. The most commonly used model is the Arrhenius equation, which correlates temperature and rate of reaction to predict the degradation rate at lower temperature. Then the real-time data analysis. Statistical tools such as regression analysis and probabilistic modeling are used to analyze real-time stability data. These methods help estimate shelf life and identify trends in degradation patterns over extended period. Conclusion on methods and techniques for stability testing. Each of the methods and techniques described above provide crucial information for assessing the stability of pharmaceutical products by employing a combination of physical, chemical, microbiology, and statistical methods. Stability testing ensures that drug products meet quality standards throughout their shelf life. The use of multiple techniques allows for a thorough understanding of how different factors such as temperature, light, humidity, and microbial combinations affect the product's stability. Then we have the regulatory requirements for stability testing. Stability testing is a critical component of the drug development process and is heavily regulated by agencies to ensure that pharmaceutical products maintain their safety, efficacy, and quality throughout their shelf life. Regulatory authorities such as the US Food and Drug Administration, European Medicinal Agency, and World Health Organization have established guidelines for conducting stability studies. These guidelines aim to standardize the processes and ensure that drugs meet the requirement standard for commercialization. One of the guidelines is Guidelines for Stability Testing. The guideline provided by regulatory agency varies depending on the region, but they generally align on several key points, including the types of stability studies required, the testing conditions, and method for evaluating the data. Some of the most important guidelines include the International Council of Harmonization. The guideline is the ICH is an organization that brings together regulatory authorities from the United States, Europe, and Japan to establish global standards for the development and registration of pharmaceutics. The ICH stability testing of new drug substance and product guidelines are among the most widely followed worldwide. These guidelines recommended recommend conducting stability studies under three primary conditions: long-term, which is the real-time stability testing, accelerated stability testing, and the intermediate condition. The long-term refers to the testing conducted under standard climatic conditions such as 25 degrees Celsius with 60% relative humidity for a period of 12 months or more. And the accelerated stability tests are typically conducted at higher temperature, which is at 40 degrees Celsius and 75% relative humidity for a shorter period of 6 months, allowing the predictions on the shelf life. Then the intermediate testing involves conditions that are intermediate between the long-term and the accelerated test, and is typically conducted at 30 degrees Celsius with 65% relative humidity. According to ICH guidelines, the stability study should also assess both the drug product and its components, like excipients, packaging materials, to ensure the formulation is stable over time. Then we have the US FDA guidelines. The US FDA also has specific requirements for stability testing in its guidelines for industry. Stability testing of drug substances and drug products. These guidelines follow the same general principles as ICH, but also include additional requirements specific to the US market. The FDA recommends conducting stability studies on the drug in its final packaging and evaluating the stability of the product under conditions that reflect both intended storage conditions and extreme stress conditions like temperature, variation, humidity, and light exposure. Now, the European Medicine Agency, their guidelines. Similar to ICH and FDA guidelines, the European Medicine Agency stability testing of active substances and medicinal products also emphasizes the importance of performing real-time accelerated and intermediate stability studies. The European Medicine Agency guidelines specifically stress the need to assess the stability of both active pharmaceutical ingredients and the drug products, accounting for packaging and storage conditions. The role of packaging in stability testing. That's another regulatory. So, packaging plays an essential role in preserving the stability of pharmaceutical products. Regulatory agencies require stability testing to be conducted with the drug product in its final packaging to ensure that the packaging materials do not interact negatively with the product or degrade over time. The container closure system, which includes the primary packaging material like the bottle, vials, and blitters, is crucial for maintaining the stability of the drug. Packaging materials are tested for compatibility with the drug product to ensure they do not cause degradation through leaching, absorption, or chemical interactions. Additionally, packaging helps protect the drug from environmental factors such as moisture, oxygen, and light. The regulatory guidelines specify the type of material that should be used in packaging for different types of drug formulations, like liquid or solid dosage form. Stability testing Stability testing for biologic products includes vaccines, monoclonal antibodies, and gene therapies. Have unique stability testing requirements due to their complex nature. Unlike traditional chemical drugs, biologics can be sensitive to temperature, fluctuation, shear stress, and pH changes. Regulatory bodies such as FDA and EMA have specific guidelines for stability testing of biologics that include tests for three things. Which one of them is protein integrity? Biologics often contain proteins which are sensitive to temperature, pH, and agitation. Stability testing for biologics often include assessing protein conformation, aggregation, and potency. Then storage condition. The biologic often require cold storage condition at 2 to 8 degrees Celsius or even below freezing. And stability studies must assess the impact of storage condition on their stability. We also have sterility. Since biologics are often administered parenterally, sterility testing is mandatory to ensure that no microbial contamination occurs during storage and handling. Stability testing for different dosage forms. The type of pharmaceutical dosage form significantly influence the stability testing approach. Different formulation may degrade in various ways due to their unique properties. For example, solid dosage form like tablets, capsule. These are generally stable, but factors like humidity, temperature, and light can affect their integrity. Stability testing for solid dosage forms typically focuses on ensuring the active ingredient remains intact and the product does not break down over time. Then liquid dosage forms, suspensions like solutions, their formulations are more prone to degradation due to the presence of solvents, preservatives, and the possibility of microbial contamination. Stability testing for liquid dosage form often include testing for pH changes, microbial contamination, and the stability of the drug ingredients in the solution. Then we have the topical and injectable products. For products like creams, ointments, and injectable formulations, stability testing is performed to assess the physical and chemical change in the formulation and ensure sterility. For injections, testing for endotoxins and particulate matter is critical. Now we have the fifth one to be stability data and shelf life determination. Once stability testing is complete, regulatory agencies require manufacturers to submit data on the shelf life of the drug product. Shelf life refers to the period during which the drug remains within the approved specification for safety, efficacy, and quality when stored under recommended conditions. Data from stability studies include physical, chemical, and microbiological testing are used to support the shelf life claims. If the product undergoes any significant degradation, the shelf life may need to be adjusted or the drug may not be approved for market release. Regulatory authorities use this data to determine whether the drug meets the necessary requirements for approval and whether additional post-market stability monitoring is required. Conclusion on regulatory requirements for stability testing. Regulatory requirements for stability testing are comprehensive and designed to ensure that pharmaceutical products are safe, effective, and of high quality throughout their shelf life. These guidelines set by agencies such as ICH, FDA, EMA, and WHO provide a standardized framework for conducting stability studies, but submitting the required data to regulatory bodies by adhering to these regulations, pharmaceutical manufacturers can ensure that their products meet the necessary quality standards and maintain their safety and efficacy from production through to consumers. Challenges in product stability testing. Stability testing is essential for ensuring that pharmaceutical products maintain their quality, safety, and efficacy throughout their shelf life. However, this process is not without challenges. Below are some of the major difficulties faced during stability testing. One is variability in environmental conditions. Stability testing involves exposing pharmaceutical products to various environmental conditions such as temperature, humidity, and light. However, these factors can vary significantly depending on the geographical region and storage conditions. Variability in storage environment can affect the product stability, leading to inconsistent results across different regions. The variability includes 1. Temperature fluctuation. The stability of a drug is highly dependent on temperature. Extreme fluctuations such as temperature spikes during transport or storage can lead to degradation of active ingredients. Regulatory guidelines specify temperature conditions to be at least 25 degrees Celsius for long-term storage and 40 degrees Celsius for accelerated testing, but real-world storage conditions can vary significantly. Another reason for inconsistent results is humidity and moisture. Moisture can degrade many pharmaceutical products, particularly solid dosage form, leading to changes in chemical composition, dissolution properties, and microbial combinations. Some dosage forms like that are highly sensitive to moisture and may disintegrate or form hygroscopic compounds. Ensuring consistent humidity levels during testing is critical to obtaining accurate data. Another factor is light exposure. Light, especially ultraviolet light, UV light, can break down certain active ingredients in drugs. Many drugs are photosensitive, and exposure to light can cause degradation. Stability testing must simulate conditions where products might be exposed to natural or artificial light sources, particularly for products with high potential for light-induced radiation, like antibiotics or biologics. Limited duration of stability studies. One of the biggest challenges in stability testing is that real-time stability studies require extended time frame, often several months or years, for new drug products. This presents a problem as manufacturers need to make shelf life predictions quickly for regulatory approval, especially for products intended for mass market consumption, which includes accelerated stability testing. Accelerated testing involves exposing the product to extreme conditions in a shortened period to predict long-term stability. While this can provide useful data quickly, it may not always be accurate, as not all degradation pathways can be accurately modeled in accelerated conditions. For example, accelerated testing may not stimulate the effect of slow chemical degradation under typical storage conditions. Then we have long-term testing cost and time. Real-time stability testing can be expensive, requiring ongoing monitoring and testing of batches over extended periods. The time and cost involved in conducting these studies can be bothersome, especially for small or mid-sized pharmaceutical companies. So, we also have complexity of biologics and biotechnology products. Biologics such as monoclonal antibodies, vaccines, and gene therapies present additional challenges in stability testing due to their complexity and sensitivity. Biologics are made from living organisms or contain biological material, which makes them more susceptible to degradation caused by environmental factors like heat, pH, and shear forces. We also have variability in packaging materials. Packaging materials play an essential role in maintaining the stability of pharmaceutical products. However, there can be significant challenges in ensuring the compatibility of packaging with the drug product. Packaging must not only protect the drug from environmental factors like light, moisture, and oxygen, but also ensure that there is no interaction with the drug that could cause degradation, which the factors could be leaching of components and barrier properties. Then we have inaccurate or incomplete data collection. Accurate and reliable data collection is essential for stability testing. However, problems may arise during testing that can affect the quality of the data. And there are two of the problems, which is inconsistent sampling and lack of standardization. Also, data interpretation. Interpret rubbish. Then we have cost and resources constraints. Stability testing can be a resource-intensive process. Large quantities of product samples are often required to conduct stability studies over extended periods. The cost of materials, labor, testing equipment, and storage can be significant, especially for small, medium-sized pharmaceutical companies. We have the testing facilities and labor and time. Testing facility conducting stability studies require specialized storage facilities that can stimulate varying temperature and humidity conditions. Labor and time. Stability studies require ongoing monitoring, testing, and analysis, which can strain human and financial resources. The time commitment involved in real-time stability studies can also be burdensome for some companies working to bring products to market quickly. Conclusion on challenges in product stability testing. While stability testing is essential for ensuring the safety, efficacy, and quality of pharmaceutical products, it is not without its challenge. Variability is in environmental conditions and the complexity of biologic products, limitations in packaging, and the high cost of testing can all complicate the stability testing process. Overcoming these challenges require careful planning, adherence to regulatory guidelines, and use of advanced techniques and technologies. By addressing these challenges, manufacturers can ensure that their products remain safe and effective throughout their shelf life. Then we have best practices for ensuring product stability. Ensuring the stability of pharmaceutical products is crucial for maintaining their several best practices can help pharmaceutical manufacturers and researchers ensure the successful long-term stability of their products. These practices encompasses everything from formulation strategies to packaging choice and storage conditions. One of the best practices is formulation development and optimization. Formulation play a central role in stability of pharmaceutical products. Proper formulation techniques can significantly extend the shelf life of a drug, even in challenging environment conditions. We also have packaging design and materials. Packaging play a crucial role in protecting pharmaceutical products from external factors such as moisture, light, and air, and microbial contamination. The choice of packaging material design should be aligned with the chemical and physical properties of the drug. Best practices for packaging include barrier properties, moisture control, child resistance and tamper evidence packaging, and compatibility with the drug. We have optimizing storage condition. Correct storage conditions are critical for maintaining the stability of pharmaceutical products, like temperature control, humidity control, light protection, and handling during distribution. We also have monitoring and testing during shelf life. Ongoing stability testing throughout their product shelf life is essential for ensuring that the drug remains effective, safe, and of high quality. And best practices in stability monitoring include real-time stability studies, accelerated stability studies, in-use stability testing, and microbial testing. Also have compliance with regulatory guidelines. Pharmaceutical manufacturers must adhere to strict regulatory guidelines when developing and conducting stability testing. We have case studies on product stability testing. Case studies play a crucial role in demonstrating the practical application of product stability testing in the pharmaceutical industry. These real-world examples provide valuable insights into the challenges faced by manufacturers and methods used to address issues related to product stability testing. The following are the highlights. Case study 1, stability of paracetamol tablets. Paracetamol is one of the most commonly used over-the-counter analgesic worldwide. A notable case study focused on the stability of paracetamol tablets, particularly in humid environments. During routine stability testing, researchers discovered that paracetamol tablets exposed to high humidity conditions experienced a significant loss of potency over a short period of time. The degradation was attributed to the moisture sensitivity of the active pharmaceutical ingredients. Solution and outcome. To address this, the manufacturer reformulated the tablet, adding moisture absorbing excipients and improving the tablet's coating to provide a better moisture barrier. Additionally, packaging was enhanced to include desiccants within each bottle, preventing exposure to humidity. Long-term stability studies under the humid conditions showed that the modified formulation retained more than 95% of its potency throughout the shelf life, meeting regulatory requirements for expiration date. Case study 2, light sensitivity of vitamin A in capsule formulation. Vitamin A is highly sensitive to light and can degrade rapidly when exposed to UV light, leading to reduced efficacy. A case study on the stability of vitamin A capsules demonstrated that exposure to light during storage could cause the active ingredient to break down, diminishing the therapeutic effect. Solution and outcome. The manufacturer responded by changing the packaging from clear plastic bottles to amber-colored glass bottles, which provided better protection from light. In addition, the capsules were stored in light-proof cartons during distribution and sales. Stability testing under various light exposure conditions showed that the modified packaging significantly reduced degradation, allowing the product to maintain its efficacy throughout the intended shelf life. Case study 3, stability of biologics during transport. Biologic products such as monoclonal antibodies and vaccines are often highly sensitive to temperature fluctuations. A case study involving the stability of a monoclonal antibodies used in cancer therapy highlighted the challenges posed by maintaining the product stability during transport. The product, which required refrigeration, was subjected to temperature excursions during transportation due to inadequate temperature monitoring system in the shipping containers. Solution and outcome. In response, the manufacturer implemented a more stringent temperature control system for transport, including the use of specialized refrigerated shipping containers equipped with real-time temperature monitoring sensor. The sensor ensured that the product remained within the recommended temperature range. Additional testing confirmed that the new transport system prevented temperature excursions and preserved the stability and potency of the monoclonal antibody, allowing it to be delivered to healthcare providers without compromising its therapeutic efficacy. Case study 4, stability of sterile injectable products. Sterile injectable products are particularly susceptible to microbial contamination and degradation, especially when exposed to improper storage conditions. A case study of a sterile injectable product used for pain management revealed that the product showed signs of microbial contamination after multiple openings of the vial during its shelf life. The product was stored in a standard glass vial with a rubber stopper. Repeated exposure to air and the handling process led to contamination. The solution and the outcome. Solution involved switching to a more advanced packaging system, a pre-filled tamper-evident syringe. The packaging minimized the risk of contamination by preventing repeated exposure to light and reducing the likelihood to microbial growth. In addition, stability studies showed that the new packaging solution extended the shelf life of the product by maintaining its sterility and potency for a longer period. The implementation of tamper evidence seals and a closed system via also contributed to the preservation of the product's integrity. Case study 5. Stability of oral solid dosage forms in high immunity regions. A case study involving oral solid dosage forms, specifically tablets, highlighted the challenges associated with storing drugs in high immunity regions. The products, a common antihypertensive medication, began to degrade when exposed to high levels of moisture during storage in tropical climate. Solutions and outcome. The manufacturer responded by reformulating the tablet to include moisture resistant excipients and increasing the use of moisture barriers in packaging. In addition, they introduced blister package in aluminum foil to provide an extra layer of protection against humidity. Stability testing conducted in various humid environments confirmed that this change resulted in a significant reduction in moisture-related degradation, allowing the products to maintain its efficacy and extend its shelf life even in humidity regions. Case study 6. Stability of topical creams in extreme temperatures. Topical creams, particularly those used for dermatological conditions, often have specific storage requirements to maintain their efficacy. A case study on the stability of a topical corticosteroid cream revealed that the product experienced phase separation and loss of potency when exposed to extreme temperatures during transportation and storage. Solution and outcome. The manufacturer introduced a temperature-sensitive packaging solution, which included insulation material to buffer temperature extremes. They also revised the formulation slightly to increase the stability of the active ingredients at higher temperature without compromising the product's therapeutic effectiveness. Stability testing under varied temperature conditions showed that the new formulation and packaging combination resulted in better product stability, even when exposed to high temperature during distribution and use in hot climates. Conclusion on case studies. These case studies underscore the importance of product stability testing in real-world scenarios and highlight the diverse challenges that manufacturers face, from light sensitivity in vitamin A capsule to the complexities of biologics transportation. Stability testing remains a crucial tool for ensuring that pharmaceutical products remain safe and effective for consumers. By learning from these case studies, pharmaceutical companies can implement improved formulation, packaging, and storage solutions to enhance product stability and ensure the highest level of quality and efficacy in their product. General conclusion. Product stability testing is an essential component of pharmaceutical product development, ensuring that drugs maintain their efficacy, safety, and quality throughout their shelf life. Stability testing evaluates the effect of various environmental factors, including temperature, humidity, light, and packaging materials, on the physical, chemical, and microbiological properties of pharmaceutical products. It helps manufacturers predict a product's longevity, determine appropriate storage conditions, and establish an accurate expiration date. The importance of stability testing cannot be overstated. It not only guarantees the product's therapeutic effectiveness, but also protects patients from potential harm caused by degraded or ineffective medications. Furthermore, stability testing plays a vital role in complying with regulatory requirements and ensuring that products meet international standards for quality assurance. As highlighted in the case studies, manufacturers must continuously address challenges posed by external factors such as the light sensitivity, temperature fluctuation, and moisture by using appropriate packaging, formulating drugs which withstand environmental conditions, and conducting comprehensive stability testing. Companies can extend the shelf life of their products while maintaining the integrity and performance. Product stability testing is a critical practice that underpins the entire life cycle of pharmaceutical products. It ensures that medication remains safe and effective, safeguarding public health and contributing to the success of the pharmaceutical industry moving forward. The evolving nature of pharmaceutical formulation and advancements in packaging technologies will continue to enhance the accuracy and reliability of stability testing, ensuring better outcome for both manufacturers and consumers. Thank you.
All right, thank you. Thank you.